For your continued presence, my name is Alexandre Gervaud and I am a high school student. This presentation discusses cortisol therapy for cancer treatment in leukemia and beyond. However, what is leukemia? Leukemia is a form of cancer that arises from the uncontrolled production of dysfunctioning myeloid or lymphoid white blood cells produced in the bone marrow, which circulate in the bloodstream. This phenomenon reduces space for healthy blood cells, reducing oxygen supply and immunity, causing fatigue, fever, unexplained weight loss, and potentially death. A current treatment against leukemia includes chemotherapy, which entails the injection of drugs potentially causing side effects such as nausea, bleeding, hair loss, and fatigue. Another is radiation therapy, employing radiation X-rays, which can harm healthy cells. Additionally, targeted therapy includes drugs and monoclonal antibodies causing side effects or even cancer resistance. On the other hand, CAR T-cell therapy could offer a more targeted approach with significant efficiency and reduced toxicity, including against relapsed leukemia. Although CAR T-cell therapy was approved relatively recently, its development has spanned several decades. Notably, the year 2011 witnessed a successful clinical trial of CD19 CAR T-cell therapy in patients with chronic lymphoblastic leukemia. In 2014, third-generation T-cells were introduced, knowing that nowadays we're at the fifth one, and with a caspase-9 gene system for regulation in case of dysfunction. Starting in 2017, the FDA has approved 7 CAR T-cell therapies for the treatment of acute lymphoblastic leukemia, relapsed diffuse large B-cell lymphoma, and B-cell precursor acute lymphoblastic leukemia, as well as relapsed or refractory mantle cell lymphoma, large B-cell lymphoma, and multiple myeloma. Chimeric antigen receptors, or CARs, have evolved, however. They all include an antigen binding domain, an hinge region, a transmembrane domain, and an endodomain, the latter of which is different for each generation as we see on the graph. The first generation contained only immunoreceptor tyrosine-based activation motifs, known as ITAMs, for T-cell activation with limited potency. The second generation added a single costimulatory domain to enhance survival. The third generation incorporated 2 costimulatory domains for stronger activation. The fourth generation was made to secrete cytokines. And finally, the fifth generation added an intracellular cytokine receptor domain to directly promote proliferation. However, how does CAR T-cell therapy work as a whole? Overall, a patient's T-cells are genetically engineered to bind to specific proteins, antigens, on cancer cells and kill them. It entails several steps. First, a patient's T-cells are removed from the patient's blood via leukapheresis. Secondly, the genes for a special receptor, called a CAR, is inserted into the T-cells in the laboratory to permit binding to specific cancer surfaces. Third, millions of CAR T-cells are then grown. Fourth, they are subsequently given to patients by intravenous infusion. And lastly, the CAR T-cells bind to cancer antigens and release substances like perforin and granzyme, once in the body, that kill the tumor cell via apoptosis when releasing cytokines. CAR cell therapy has mainly proven effective against blood cancers, including leukemia, lymphoma, characterized by abnormal lymphocyte proliferation, and myeloma, featuring plasma cell malignancy. CAR cell therapy demonstrated an impressive combined complete response rate of 92% against B-cell acute lymphoblastic leukemia, meaning that right after treatment, 92% had no detectable cancer remaining. Efficacy has been revealed in other blood cancers as well, although to a lesser extent than for leukemia. For comparison, CAR cell therapy was more efficient than the more conventional immunotherapy blinatumomab in ALL patients, with an 86% rate of complete remission versus 84% for the other drug, blinatumomab. However, CAR cell therapy faces several key challenges, despite its potential. First, on-target off-tumor effects cause CAR T-cells to attack healthy tissues that express the target antigen. Moreover, through antigen escape, tumor cells can lose the target antigen. Trafficking and infiltration issues also limit efficacy when CAR T-cells fail to reach or penetrate solid tumors due to the extracellular microenvironment. Finally, the immunosuppressive tumor microenvironment can impair CAR T-cell survival and function. The fifth and most significant side effect, however, is toxicity. The most common types of toxicity are the cytokine release syndrome, known as CRS, and immune receptor cell-associated neurotoxicity syndromes, ICANS, which are really linked to excessive cytokine production and T-cell activation. In a study, CRS occurred in 70% of leukemia patients, for example, and 93% of lymphoma patients, and severe ICANS in up to 70% and 62% respectively. The proportion of severe CRS made up only a part of this total. So, against B-cell malignancy, other toxicities were also noted, with frequencies going as high as 94% due to blood cell loss, although those were less significant. Despite restraints, CAR T-cell therapy has shown success in the combat of a variety of solid tumors. For example, Claudid 18.2 CAR T-cells achieved a 33.3% overall response rate, causing cancer shrinking in 33% of metastatic gastric cancer patients. MSLN-targeted therapy achieved 82% in 1-year survival in malignant pleural mesothelioma patients. And as another example, GD2-targeted therapy induced a complete remission rate of 27% against neuroblastoma, as seen on the left of the table. And this illustrates the increasing improvements made in the area of solid tumor treatment, although improvement can still be done. Since 2017, the Food and Drug Administration, FDA, has approved 7 CAR T-cell therapies for hematological or blood malignancies. Those are Kymriah, Yescarta, Tecartus, Breyanzi, Abecma, Carvykti, and Ocatil. Despite these milestones, median total cost exceeding $600,000 and inconsistent insurance coverage limit implementation. Access is further restrained by weeks of manufacturing time, limited treatment centers, state Medicaid restrictions, and complex billing processes. Next-generation CAR-based therapies could overcome some of these deficiencies. Innovations include bispecific AND gates for greater specificity, cytokine modulation to boost persistence, transcription factors tuning to prevent exhaustion, CRISPR RNA editing for precise genetic control, CAR macrophages or core macrophages for direct tumor phagocytosis, in vitro transformed mRNA CARs for controlled short-term activity, organoid testing for precision targeting, artificial intelligence for toxicity prediction, and in vivo CAR generation to bypass ex vivo manufacturing. And we could add that nanobody-based CARs can also improve stability. Other innovations include allogeneic CAR therapies, which use immune cells from the healthy donors to create off-the-shelf cancer treatments, available for immediate use for other patients. Allogeneic products can be produced in large batches, lowering costs and enabling rapid and accessible treatment. Different cell types exist for such treatments. Alpha-beta T cells to offer potent toxicity, natural killer and gamma delta T cells to provide MHC-independent tumor targeting, iNKT cells that bridge innate and adaptive immunity, memory T cells to offer long-term persistence, and virus-specific T cells that combine antiviral and anti-tumor functions. In essence, CAR cell therapy represents a revolutionary personalized cancer treatment that has transformed outcomes in hematological malignancies, so specifically for leukemia, lymphoma, and myeloma. However, challenges persist, ranging from severe toxicities, antigen escape, and infiltration difficulties to manufacturing delays, high cost, and limited availability. Ongoing developments, including dual targeting CARs, cytokine modulation, and allogeneic products, aim to overcome these barriers, showing promise for solid tumor treatment that is still being developed and researched. But with continued research, CAR cell therapy could become a safe and efficient cornerstone of cancer treatment, becoming even more efficient and safe than others like chemotherapy and radiation therapy. Here are the references of my visuals and studies. And finally, I would like to thank Dr. Kim-Marie Dam for her guidance and mentorship as my mentor throughout this Polygence project, as well as the Polygence platform itself. And I also wish to express my gratitude to my family for its support. And finally, thank you for listening.
Great job. Thank you so much. I really like your the figures that you used throughout too to really demonstrate the points that you were making. One of the questions I had in mind was like, what is the difference between the different generations? And you that figure really made it super clear, and you walked that through really clearly. So that stood out to me as really great. Cool. We have a question from the Q&A before I get into others. Um, this question is about leukemia specifically. Who's at the highest risk of developing leukemia?
It depends on the leukemia type, but for example, for acute lymphoblastic leukemia, also known as ALL, the most affected persons are children. Generally, yeah, children or to adolescents, maybe young adults, but not really beyond, or at least for the most part. However, for other ones, like BAAL, those can be for much older adults, like young people, especially white males and white individuals in general, especially in the American population. But I would say that except for AAL and some other exceptions, in general, leukemia types are more frequent in older adults.
Super interesting. And I think that's one thing that stood out to me too, the leukemia, just that bar graph where it was so much more effective for leukemia than other cancer types. And what is the thinking behind that? Why is CAR T treatment so much more effective for leukemia?
Can be more effective than other treatments, such as radiation therapy, for example, because CAR T therapy aims to really um, modify the T cells that act as the immune system soldiers in the body to equip them with the proper equipment to really target specifically the cancer so that it binds to the cancer, attacks them, and releases the specific substances such as granzyme and porphyrin to grab the cell, transfers them and kill them. While, for example, radiation therapy is less specific and it's just a shooting of rays to try to kill them, but without really focusing on cancers and even also damaging other parts of the human body. So
Yeah.
in addition to the efficacy due to the specific binding that is genetically modified, there also is the component of specificity. Although um, researchers are still trying to find um, receptors that are specific to cancers, because sometimes receptors are both related to cancers and human body regular cells. So sometimes the toxicities come from there, from the bivalence of the receptors. So it is still developing, but as I have discussed, it has shown significant results like this.
Awesome. Great job. And then what about you? What do you want to do next? What's your hopes and dreams for what's gonna happen to you next?
I am a high school senior, and I plan to major in biology in college to then do research and try to improve the scientific field and explore more, to eventually potentially become a pre-med student or try to enter a medical school. But in the shorter term, I think I will focus on research and exploring advanced scientific courses, and including in immunology, which interests me, as well as oncology.
Great. Yeah, super interesting. And you're on the right track for all of that now. So it's all in your future for you for sure.